This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is a promoter? Answer: a promoter is a site where RNA polymerase II and multiple other transcription factors bind to DNA upstream from the gene locus. It typically includes an at-rich upstream sequence with TATA and CAAT boxes, which differ between eukaryotes and prokaryotes. A promoter mutation commonly results in a dramatic decrease in the level of gene transcription. Question: how do enhancers work in relation to gene expression? Answer: enhancers are DNA loci where regulatory proteins bind, increasing expression of a gene on the same chromosome. Enhancers and silencers may be located close to, far from, or even within the gene whose expression they regulate. Question: how do the TATA and CAAT boxes differ between eukaryotes and prokaryotes? Answer: the TATA and CAAT boxes that are found in the upstream sequence of a promoter are different between eukaryotes and prokaryotes. Question: what happens when a promoter mutation occurs? Answer: a promoter mutation commonly results in a dramatic decrease in the level of gene transcription. Question: how does the location of enhancer affect gene expression? Answer: enhancers and silencers may be located close to, far from, or even within the gene whose expression they regulate. This means that the location of enhancer can affect gene expression. Question: can you give an example of how enhancer increases gene expression? Answer: an enhancer is a DNA locus where regulatory proteins bind. By binding to the enhancer site, the activator proteins increase the expression of a gene on the same chromosome. Question: are enhancers and silencers always located close to the gene whose expression they regulate? Answer: no, enhancers and silencers may be located close to, far from, or even within the gene whose expression they regulate. Question: how do enhancers differ from promoters? Answer: promoters are the site where RNA polymerase II and multiple other transcription factors bind to DNA upstream from the gene locus, while enhancers are DNA loci where regulatory proteins bind to increase expression of a gene on the same chromosome. Question: is there any overlap between enhancers and silencers in terms of location? Answer: enhancers and silencers may be located in the same general region, but they have distinct functions in regulating gene expression. Enhancers increase gene expression while silencers decrease gene expression. Question: are enhancers only found in eukaryotes? Answer: enhancers are found in eukaryotes, but not in prokaryotes. Question: can enhancers also be called as activators? Answer: yes, enhancers are also known as activators as they bind to the enhancer site and increase the expression of a gene on the same chromosome. Question: can you give a real-world example of how a mutation in an enhancer can impact gene expression? Answer: a mutation in an enhancer can impact gene expression by disrupting the binding of activator proteins to the enhancer site. This can lead to a decrease in the expression of the gene that the enhancer is regulating. For example, some genetic diseases such as cancer are caused by mutations in enhancer regions that lead to abnormal gene expression. Question: can an enhancer be located within an intron? Answer: yes, enhancers may be located within an intron. Question: are there different types of enhancers? Answer: yes, there are different types of enhancers, such as tissue-specific enhancers, which are active in specific cell types, and constitutive enhancers, which are active in all cell types. Question: are there any other factors that can affect the binding of activator proteins to enhancers? Answer: yes, there are many other factors that can affect the binding of activator proteins to enhancers, such as chromatin structure, epigenetic modifications, and the presence of other proteins.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.